Alright, so today, two topics we're going to cover, which the first one is polygenic traits, and for those people that have done the lab on where you made the offspring, you came across polygenic traits when you were talking about eye color, hair color, and skin color. Polygenic traits is defined as any trait that's going to be controlled by more than one gene. And the majority of the times you see things, here is an example of eye color. And if you notice, it has A's, B's, and C's. Each one of the different letters signifies a different gene. There are two copies of each gene. So for have dark brown in terms of eyes, you see it's got a homozygous state dominant-wise for A, B, and C. On a test or something, you'll be given a scale with a specific phenotype matching maybe one genotype. There are many other genotypes that aren't represented here, but what you look for is the trend. And the trend for dark brown is that you see 6 dominant alleles. Then you go down to medium brown and you identify that there are 5. So it starts going down by 1 each time until you get all the way down to blue, which is the homozygous recessive state. And from this point, if I had thrown a random genotype at you that was potentially something like big A, big B, little c, this is not represented on the scale over here, so some people will be confused saying, I can't predict what phenotype that is. To make it as simple as possible, textbooks will just count the number of dominants. So this one has 1, 2, 3 dominants, so it will correspond to the scale here that has 3 dominants. It's the hazel. So hazel has 1, 2, 3 according to this diagram, so therefore this genotype will also match the hazel. Alright. For this one, it comes out to be a simple tri-hybrid cross. So I'm going to do that on the next example, the next slide. So I'm going to pick 2 phenotypes from right here. Let's say that a person that has green eyes is going to mate with a light brown. So I'm going to go back and forth because I won't remember this all at once. So we have big A, little b, big C was the green, and the light brown was big A, big B, big C. So now this is simply a tri-hybrid cross. You'll come out with the gametes. This parent on the left would produce a dominant A, recessive B, dominant C as one variation, another variation, dominant A, recessive B, recessive C as a second, then recessive, dominant, recessive. So this parent has 4 combinations. Then this parent also will have 4. I'm going to bypass and go right along the second. Pretend this is the top of the Punnett square, and this is going to be the side now. So the first combination this parent can produce, dominant, recessive, dominant, and finally recessive, dominant. So this is how you would have completed that tri-hybrid. You could fill it in like it was a true tri-hybrid, where you would do big A, big B, little b, big C. But technically we're not looking at this full genotype. All I really care about is the number of dominants. So to simplify this, to make it easier for you and a little less confusing, I'll keep that one there, but really all I care about to figure out the phenotypic ratio is the number of dominants. There are 5 in this box. That's really what I care about. In this box, that's all I'm going to do. I'm going to put the number of dominants that you would have combining this gamete with this gamete. So this one would bring 3, this one would bring down 1. So I'm going to put a 4 in that box because I really don't care about the genotype. I'll continue that across the board. This one still has 3, this one has 1, so there's another 4 dominants that would be placed in that square. This one 3, this one has 0. So now that one is only have 3 from this parent. And that's how you'll complete this Punnett square. You'll have less to mess up potentially because you won't have as many letters to confuse yourself. So here is 2 plus another 2, so it's 4. So I'm not going to fill the rest of this in, but you get the idea of what's happening. When this is completed, I then will go back to my scale and find out how many of the ones that had 4 were there. So I knew that I had a lot of light browns already because I had, from the Punnett square, I had 1, 2, 3 already that had 4 dominants. So I know my ratio is going to have some light browns. There was one already that had 5 dominants in it, so they potentially, with the cross that I made, could have a medium brown-eyed child. And that's all you do. That's a polygenic. If there are 4 genes, like maybe skin color, eye color, you would have a 4th gene, probably letter D, and then even more combinations of skin color and skin tones. But you do the same thing that you would do with a trihybrid, now you do with 4 genes coming with combinations. So that's an example. If you have more questions about polygenic, you can ask me that tomorrow. The second thing that we're going to look at today is something called a sex-linked trait. To this point, you have been looking at, if this picture is called a karyotype, it's a combination of all of your chromosomes within one cell. Because this is a human chromosome karyotype, there are 23 pairs of chromosomes. Remember that idea of diploid number being 46 and haploid being 23? So what they've done is they matched up all the homologous pairs together. They're right next to one another on both of these. The only thing that's different between these two is the last pair. They have 1 through 22, and then they have this pair. If you can see this, these two homologous matches are pretty similar in size because they're both X's. That makes this karyotype that of a female. This karyotype, once again, numbers it from 1 to 22. They actually go in size order. The biggest chromosomes are 1, the smallest chromosomes are 22, and it descends in size from that point. The last pair is one is larger than the other because the Y chromosome is a lot smaller. It has less numbers of genes than the X chromosome. So this is an XY combination, which makes it a male. To this point, we've been looking at all genes found on 1 through 22. If I covered this up and I covered this up, you would not be able to tell the gender of this person. The only thing that designates gender is the combination that happens right at the end, the X and the Y, or the X and the X. So if I found a gene that was on chromosome number 15, right here, both males and females have 2 copies of that because they have 2 copies of chromosome number 15. So when you do a normal cross, that's what you've been doing. Big A crossed with big A, little a. Because on chromosome 15, both the female and the male have 2 copies of that chromosome. But what you're learning today is that sex-linked problems is any gene found on a sex chromosome. There are 2 types of sex chromosomes, the X and the Y. We don't focus really on any sex chromosome or sex-linked genes that are found on the Y. We really focus on sex-linked chromosomes that are found on the X chromosome. So notice here on the female karyotype, there are 2 X's. On the male, they only have 1. So the genotypes are going to be slightly different, and the way they inherit these genes will be different based on your gender, whether you're a male or a female. There are 3 types of disorders that you need to memorize that are sex-linked. The first one is colorblindness. The second one is muscular dystrophy. And the third one is hemophilia. All 3 of these are X-linked. That's what is another way to say it. Means the gene for hemophilia is found on the X chromosome, not on the Y chromosome. So all 3 of these are all found on the X chromosome. So to set up an actual Punnett square or a genotype for a sex-linked problem, you first set up the chromosome combination of the female, then you set up the male. So I told you when you were doing codominance you have to avoid I, X's, and Y's because the notation of sex-linked is going to mimic that of codominance, but they're not necessarily, they don't act the same way. If you are a female, you have 2 X's, so therefore you have 2 locations of where the gene can be located. For the male, they only have 1 X. So they get one copy of that gene because the Y chromosome, which is the other homologous match, doesn't get that gene. So there is no colorblindness gene here. Males will only inherit one copy of either the normal form of colorblindness, which is, what we'll say, it's the dominant form, or they get the recessive. If I tell you that a female that is a homozygous normal person for color combination, what you do is you do X, it's like the superscript of codominance, with two capital C's. Capital C represents the normal vision, you can see color. If I cross that with a male, maybe the male has colorblindness. They only get one allele because they only have one X chromosome. So this is what it would look like. This is a homozygous normal. This is a colorblind male. So if I set this up as a Punnett square, what you would do for the female, you have to be very clear with your superscripts not to mess up dominant versus recessive. You separate. The male, I said, was colorblind. So there is the recessive, and he gets the Y. Notice no letter. Never put a letter on the Y. You combine them. And this is what you get. The last thing you need to know is how to phenotypically separate them. When these are written out phenotypically, because you know the gender, you have to separate your phenotypes based on gender. So if I look at the males, there are two sets of males. Both of them are normal vision. So you can say 100% of males are normal. For the females, they have a dominant colorblind gene and they have a recessive, so they would still see normal vision. So in this case, the females are also normal. But you still have to separate out the genders for it. Okay? You would do this for any one of them. If I said that the female, the word heterozygous is not often used. The word carrier is more often used when it comes to sex-linked problems. So this is a carrier female. She doesn't exhibit colorblindness, but she has the ability to pass it on. The male, I'll say in this case, also maybe he doesn't have colorblindness, so that's what his genotype would be. Complete a Punnett square that looks like that and figure out the phenotypic ratio. So tomorrow in class, I'll make sure that this is understood a little bit better, but I wanted you to have a little bit of knowledge, a little background for tomorrow. All right? I'll see you then.